Hello, so the next little part then on cells is diffusion. What? So diffusion then. How exciting. So this shows diffusion. So what you'll notice there is that the particles in here start off where there's lots of them or where they're in a high concentration, and they move about all over the place to areas where they are in low concentration. And that's what diffusion is. Diffusion is when particles move randomly from an area of high concentration to an area of low concentration, sometimes through a selectively permeable membrane, which we'll talk about later. So if we just look at this, here is a little plant cell, and there's this little cell wall. So what's going to happen is the particles are going to diffuse from an area of high concentration, which is outside the cell, which is outside the cell, to an area of low concentration, which is inside the cell. This is just a little animation showing that. So we've got the membrane around the outside. These little circles are oxygen molecules, and we can see them diffusing from an area of high concentration into the cell where they're in low concentration. So if we're looking at this, here we have just a little animal cell. Here we have some little particles. Which way is diffusion going to occur here? Remember, it's always high to low. So we're going to move from outside to inside. This one, there's a high concentration here, a low concentration here, so they diffuse outwards. And the final one then, what happens here? Will diffusion occur in any direction? Well, you've got to remember that diffusion is random. So actually, some particles will diffuse out, some will diffuse in, and what we say is that there would be no net exchange. So what we've looked at in these examples is which way the overall diffusion will happen. Now, diffusion always happens from high to low. But because these particles don't think about where they're going, they can't really control where they're going, they do just diffuse from high to low concentration, it means that most of the particles will move this way. But some of these little particles can also move out, and that's just because diffusion is random. So overall, when we get an example like this, where the concentration is equal inside and outside the cell, some will diffuse out, but some will also diffuse in. So we say that there is no net exchange. Now, it's safe to say that in lots of living things, diffusion happens all the time. If you think about just sitting there right now, diffusion is happening when you breathe in. The oxygen diffuses from the air in your lungs into your blood. It then travels around your body, and the oxygen diffuses from your blood to the cells, which can then be used for respiration. So diffusion happens all the time. Moving on a bit from that, there's several factors that affect the rate of diffusion, and these are shown below. The three factors that affect the rate of diffusion are concentration gradient, temperature, and surface area. Now, a concentration gradient is the difference between concentration between the high and the low areas, and that's called a concentration gradient. The larger the difference, the faster the rate of diffusion. Now, if we look at this example, in this example we said that the particles were going to move from high concentration to low concentration. So where does the gradient come into that? Well, let's say, for example, we had a little graph, and here was concentration. Here was inside the cell, and here was outside the cell. Inside the cell, we can see that there's a very high concentration. Outside the cell, we can see that there's very low concentration. So, in this case, if we joined up those 2 points, this would be our concentration gradient, which is the difference in concentration between the areas of high concentration and the areas of low concentration. Now, what we're saying here is the bigger the difference between high and low concentration, the faster the rate of diffusion. So, the steeper the gradient, the faster the diffusion. Now, we can also say that temperature affects the rate of diffusion, because if you've got higher temperatures, it increases the kinetic energy of the molecules, which basically means that they move about much faster, and that increases the rate of diffusion. So, higher temperature equals faster diffusion. And then finally, surface area. So the larger the surface area over which diffusion takes place, the faster diffusion occurs. So if we want diffusion to happen quickly, here's the three things that we need. We need a steep concentration gradient, we want a high temperature, and we want a large surface area. Those three things will make diffusion happen really quickly. Now, what are some examples of diffusion? Diffusion of oxygen from the air spaces in the lungs, so the little alveoli. So this is an alveolus, and this is a blood vessel. When we breathe in, oxygen comes into here and it diffuses from an area of high concentration to low concentration. Likewise, carbon dioxide will diffuse the other way. So, another little alveolus, another little blood vessel. If we're looking at carbon dioxide, there will be a high concentration in the blood, a low concentration here, so the carbon dioxide diffuses from an area of high concentration to low concentration, so we can breathe it out. Diffusion also happens during digestion. If this is the ileum, which is the small intestine, its main job is to absorb food. One of the things about the ileum is that it has a really good blood supply, which is actually very close to the walls of the ileum. So if these are little food molecules, here they're in high concentration, and they diffuse from here, which is an ileum, to the blood where they're in low concentration. Now this part just shows us some other examples of where we have cells with a large surface area. So this is a root hair cell. It's not found in your hair or in your scalp. It's found on plant roots. And it has this little bit because it increases the surface area so that the plants can absorb more water and minerals. This part shows the ileum. These little things here, the wee bumpy things, those are villi, and they also increase the surface area. This is a red blood cell, and it has a biconcave shape, which means that it has a larger surface area so it can hold more oxygen. And this part here shows an alveolus, and because of its rounded shape like this, it has a large surface area so we can get more oxygen into the blood. Now, in the last part, we said that sometimes diffusion takes place through selectively permeable membranes. Now, permeable lets things pass through it. So we said that the cell membrane was permeable, but we also said that it was selectively permeable. So it chooses what things that it allows to pass through it. So this little experiment demonstrates that. So we get a length of dialysis tubing, which behaves like a cell membrane. The reason it behaves like a cell membrane is because it is slightly permeable. So it has these tiny microscopic holes in it, which substances can pass through or out. It's not quite as complex as a cell membrane, but it gives us a good likeness for this experiment. So what you have to do was tie one knot at the end of it and leave the other end open. You then fill the dialysis tubing with starch and rinse the outer side of the tubing to remove any traces of starch. You place the tubing with the starch into a clean boiling tube and fold the end over the tubing of the boiling tube with an elastic band. You then fill the boiling tube with dilute iodine and leave standing in a beaker for 5 minutes. So this is what you get. You get a boiling tube with some dialysis tubing in it. Which is basically like a giant suite here. Then I'm going to draw this black. Inside this you have starch, and here you have iodine. Now the iodine is dilute, but it's still yellow-brown in colour. The starch is just a colourless solution here. And this is the viscing tubing, which, remember, has the little holes in it and is semi-permeable. So you set this up and you leave it for 10 minutes and you see if there's any changes that take place. Now when you leave it for 10 minutes, this happens. The iodine changes to blue-black. Now, why did this happen? Well, first of all, you might remember that the test for starch is iodine, and iodine will change from yellow-brown to blue-black if starch is present. So somehow the iodine, which was around the outside, has moved in here. But this is strange. Why has it gone blue-black on the inside and not the outside? Well, here's why. The starch is too big to pass through the little pores in the tubing. But the iodine molecules can fit, so they diffuse into the tubing. So they diffuse from an area where they are in high concentration to an area where they're in low concentration. When the starch and iodine is mixed, it goes blue-black. So the iodine diffuses in, and then when the iodine diffuses in, the starch starts to change colour. And that's why it goes blue-black. Now this also shows a little bit about permeability. Because these starch molecules were too big, they couldn't fit out there, but the iodine molecules are nice and small, so they could fit in. Now the next little part is a tricky concept. So what effect does surface area and volume have on diffusion? So as we've said, the rate at which substances diffuse into and out of the cell depends on several factors. We said concentration gradient, temperature, and surface area. So some other things that affect it is the surface area of the cell and its volume. So the diagram below shows how surface area and volume changes as a cell becomes larger. So we're using these little cubes here to represent cells. So this is our little cell. Now, each time the cell doubles in size, what we can see is that the surface area increases each time. We can also see that the volume increases, but the volume actually increases faster than the surface area. So here's what you need to take away from this little area. The larger the cell volume, the greater the amount of substances, such as oxygen, the cell needs for its reactions. Which makes sense, because if you think about it, bigger animals need more food. So it makes sense that if cells get bigger, they need more substances. The next part then, the larger the surface area, the greater the amount of oxygen that can diffuse into the cell. Which again makes sense, because if you've only got a cell this size, oxygen will diffuse in there. Whereas if you've got a larger cell, you've got way more space where oxygen can diffuse in. Although both volume and surface area increase with size, the volume increases faster. This means the surface area available for oxygen for each millimetre of cell volume halves each time the cell doubles in size. So eventually, because the cells get so much bigger each time, eventually the cell will become so large that there is not enough surface area to allow sufficient diffusion of oxygen for the cell's needs, and it will die. So therefore, there is a limit on the size of single-celled organisms. They can only ever be microscopic, because if the cells get too big, they will die because they can't get enough oxygen. So what you need to take away from this is, as cells get bigger, surface area increases, volume increases, but volume increases more than surface area. So therefore, there is a limit on the size of single cell organisms. For example, bacteria or fungi. And that's what you need to take away from that section. Multicellular organisms are a bit different. So this is any organism which contains more than one cell. So it could be things like seaweed, a mouse, a frog, a human, basically any animal with more than one cell. So most organisms have become multicelled, making them much larger than single cells. However, as previously described, organisms need a large enough surface area to volume ratio to be able to efficiently exchange substances with their environment. Large, active, multicelled organisms have developed special gas exchange organs inside their body, for example the lungs, which have a greatly increased surface area. But this creates another problem. The gas exchange surface is often far away from many of the other body cells, so a transport system, such as the circulatory system, therefore has developed in many organisms. So what this means is, say for example, we've got a little human who's a bit blob looking. The problem is the cells on the inside of a human's body, like here for example, are too far away from its outer surface. So those cells can't rely on diffusion. So what larger animals have to have is a gas exchange system, which in our case is our lungs. So then they breathe in, and that's how they get the oxygen into their body. But the problem with that is it only gets oxygen to their lungs. So they then need to develop a specialized transport system, such as your circulatory system, which can then transport all the oxygen to all parts of the body. Now, just to finish this section off, I think we may as well do a little question. So the diagram shows an experiment to investigate the effect of increasing the concentration of iodine solution on the rate at which iodine diffuses through agar jelly. So here we have a Petri dish containing 1% iodine solution and 2% starch. Petri dish B contains 5% iodine solution. So there's the difference. You leave them for 30 minutes at 20 degrees C and you see what happens. So what we need to do is describe and explain the conclusions that can be made from the results of this experiment. So what we can see is a dark area. So here's what we can say about it. So first of all, the dark area is blue-black, and it forms because the iodine diffuses through the starch. The second thing I would say is, in B, the dark area is bigger, and that's because there is a higher concentration of iodine. Now, the thing that we need to do to describe that is there is a steeper concentration gradient. So there is faster diffusion. And the final thing is, anytime you are saying anything about diffusion, describe what diffusion is. Diffusion is the movement of particles from an area of high concentration to low concentration. And that's how you would answer questions about diffusion. Most of the time, if you're answering a question about diffusion, part of the mark scheme will be looking for you to explain and show that you know what diffusion is. And that's the end of this video. Thank you. Goodbye.